Thank you for the introduction. Can everyone hear me?
Yeah, very good.
Can you see my screen?
Yes, very good.
Okay, all right. So let me see, how do I, okay. Yeah, so I want to start by, flashing on this kind of a complicated slide. And I want to start also with a story. So when I was your age, the age of, I guess many of the audience, many of you in the audience, a long time ago in ancient times. So my kids are about your age, and that's what they call me. So at that time, I was very interested in physics. I love physics. In high school, when there were physics homework, they were fun. They were not homework. But when I was trying to decide what subject to get into when I was entering college, I decided I'm not going to study physics because I felt like everything is known. Everything makes sense already. I chose biology because nothing makes sense. It just seems so complicated and so many mysteries that I have no answer for. And I felt like it's something that I can easily spend the rest of my life studying and still finding new things. So that was really drove me into biology. What is really interesting to me, which something that I didn't imagine at the time, is that actually life comes a full circle. And it turns out that there's a ton of physics in biology. And that is where a lot of my research, as well as research at the Mechanobiology Institute, is really about. So that is about how physical properties or physical signals define biological functions. And you can imagine physics everywhere. So just a simple example, blood. So you have blood cells that make up the blood. And blood has a certain physical properties. And when these properties change, either that could be due to physiological processes such as blood clotting, or there are also pathological processes that could be very devastating. There's also bone structures. There's hard bones, soft bones, cartilages, and they're made up of different kinds of cells. And yet these cells can generate structures with very different physical properties. And the examples of physics and also mechanics in biological systems is really everywhere, from molecular scales to scales of tissues and organs. So on the molecular scale, which is nanometer scale, for example, packaging of DNA, the blueprint of our genetic code has to be packaged into chromosomes. So these are structures that allow proper interpretation or expression of the genetic information, and also allow accurate transmission of the genetic information from mother cell into daughter cells. And so this compaction process is very much a mechanical process. And then when genes are being expressed, they have to unpack locally and transiently. And again, those are very much mechanical processes that cannot go wrong. If they go wrong, that's when cells can possibly either misinterpret genetic information or even cause damage to the genome. On the cellular level, so now this is micron scale, what you're looking at is a white blood cell that's chasing a bacteria. And it's not hard to see that this is very much a mechanical process. not only the cells can move, it also can very tenaciously chasing this bacteria until at some point it engulfs the bacteria, eat it up. Okay, so that's our body's defense mechanism. But in fact, not only just immune cells can do this, many cells in our body, especially during the development of the organism or embryos, there's a huge amount of cell migration to different sites to form tissues and organs. And of course, on the even larger scale, the organs. So here you're looking at the heart and the vascular system. The heart pumps, and what drives the pumping of the heart comes from the mechanics of cardiomyocytes, as well as the endothelial system, the blood vessel system. And in the developing of these intricate organ system, there's also a large amount of mechanical processes. So for example, the heart has to loop in a certain way in order to generate the right structure and the right shape. So mechanics and physics is abound in biology. Cells and tissues not only perform their functions, through mechanics, they can also sense the mechanical properties, connect with the environment by sensing the mechanical properties. So here's a very simple example. If we put a cell in on a soft gel substrate, they form this balled up shape. And if we put cell on a stiff substrate, they nicely spread out. So, and they form these, what you see here, these yellow structures are nice connections between the cell surface with the substrate, which we call extracellular matrix. And so this shows that the cells can actually change shape in response to the mechanical environment. But in fact, it's not just the shape. Actually, cells change their gene expression program. They're actually doing different things when they are on different substrates with different mechanical properties. And cells also can choose what kind of mechanical properties they like to move into. So in this movie, what you're going to see is a cell that started off on the soft side, and then it ends up on the hard side. So cells actually like to move towards the more stiffer surface. But how does cell know that? And why do they prefer stiff surface? Those are actually questions that are not fully answered. And here's on this side, what you have is a cell that starts on the hard surface. You can see that it goes to the boundary of the between the hard and soft. It turns around because it doesn't like the soft surface. So here, what these examples shows is that cells can actually interpret cues that are mechanical, not just chemical cues, which perhaps are better known. Here, the cells actually sense mechanical cues. So that is really what mechanobiology, the subject matter of the Mechanobiology Institute, is all about. So mechanobiology is truly an interdisciplinary branch of biology because it's not just biology in the traditional sense. What is mechanobiology? We have a pretty broad definition of mechanobiology. Mechanobiology is a subject that interrogates biological systems from a physical and mechanical perspective. And this is a really new perspective to study biology because in the past several decades, the focus are mostly on the chemistry side, on the genes, on the molecules, on the proteins, on the metabolites. That is about biology as a chemical system. But what is really equally important is that biological systems is also a physical system. it has mechanics, has dynamics, and the chemistry and the physics have to be seamlessly integrated together to form cells and tissues and organs. So we are, and our research focused on the mechanical aspect. But because biology is an integrated system, we cannot depart from genes and molecules. So we have to figure out how the physics and chemistry work together. But the questions that we focus on are quite unique. So for example, force is one of the most important concepts in our research. How are forces produced to drive form and function across scales? How are mechanical input integrated with biochemical signals and even drive the generation of biochemical signals? And then in disease process, often disease processes often stem from breakage of biomechanical systems. And so we need to understand that in order to figure out how to fix it. Just like if your car stops moving, you need to figure out what are the mechanical problems, and then you can properly fix it. And so in one of the emerging area of our research is we want to understand how the biomechanical system are targeted or change during the aging process, and how we can give our mechanical systems periodic tone-ups, just like you give cars tone-ups, to prolong the healthy function and to delay aging or treat age-related diseases. So that's about what our research is about. Now I want to just give you some highlights about what are our methodologies. And so, as I mentioned, force is a very important concept that we want to understand. We want to understand how it's generated and what are its consequences. And so we need to be able to measure force. But these forces are very, very small because they come from very small objects, molecules, cells. Those are on the, like I said, nanometer, micrometer scales. And the magnitude of these forces are also very small. They're on the order of piconewton or nanonewton scale. So there are an array of advanced technologies that we employ, such as magnetic tweezers, optical tweezers, micropillar arrays, and shown here is atomic force microscopy. So these are tools that we can use to measure very, very small amount of force from very, very small objects. Just give you a example. So this is measuring cellular forces, how forces derived from a single cell using what's called a micropillar array. So these are micropillar arrays. These are very small. The diameter is just a few microns, and these pillars are flexible. Okay, so when cells crawl over them, the force exerted by the cell on these pillars cause these pillars to bend. So by imaging the amount of bending of these pillars, we can calculate the amount of forces exerted by the cell. So here's an example. You can see a cell moving over these pillars, and the pillars deflect, however minutely, but we can image and we can measure that. And therefore, we can calculate how much this migratory cell is exerting on the substrate. And these forces come from the cell's cytoskeleton filaments. And so now we can calculate how much force each of these filaments is likely to be generated, producing. So this is a very useful technology that we employ to measure cellular forces. So in addition to measuring forces, we want to see the consequence of the force. That is to drive dynamic processes again, on many scales, from molecular scale, cellular scale, to tissue, organ, and organisms, okay? And so what you're looking at here are, some of the structures. So here, this is within the cell. The blue is actually a kind of molecule called myosin that generates force. It burns burns ATP and generates energy and drives dynamics inside the cell. And here is actually, there are two cells. This cell is actually trying to eat this round cell. And here is a cluster of cells forming a mini tissue. This is actually can happen on the dish. And here is a developing embryo of the fruit fly. Okay, so here's, and we can label whether intracellular structures or specific kinds of cells with fluorescent markers. But we don't just take still images. That doesn't tell us very much. Okay, so you might have seen fixed images of dead cells, but we don't do that. We call that roadkill. We want to look at everything live, because everything is dynamic in biology. So, so what you can see is here is contraction of these muscle-like structures inside the cell. And here is actually this red cell is trying to eat this round gray cell. And here, what you're seeing is that cells are actually undergoing division. They're segregating the chromosomes, and here is the embryo that is generating a specific morphology during development. So by looking at these dynamic processes, we can also measure and deduce force and understand what might be the driving force behind these dynamic processes that generate cellular function, generate tissues, and generate embryo. So live imaging in 3 dimensions is really our bread and butter. We do that every single day. In addition to discovering, how forces are generated and how they're transmitted in biological systems, we actively apply mechanobiology principles for, translational purposes. For example, here's an example from Professor Lim's lab, Tritek Lim's lab, taking advantage of the mechanical properties of cancer cells is different. The different mechanical properties of cancer cells from the properties of other kinds of blood cell type to isolate circulating cancer cells for diagnostic purposes. Here's the principle. So there are some of these little pillars, and when normal blood cells flow through, those cells are more compliant, and they can go through these in between these pillars, whereas tumor cells are bigger and they're stiffer, so they get trapped. Here's actually that on the left side is a simulation, on this side is actually a real life movie showing trapping of a tumor cell. So this allows capture of these tumor cells, even if they're very rare in the blood. So I hope that these slides convince you that biology is not just traditional biology, that what you might be have studied in the textbook in middle and high school. Biology nowadays is really interdisciplinary science. It brings in a variety of disciplines from physics, mathematics, to chemistry and engineering. And by integrating these disciplines, we're best positioned to understand the complexity and deep mechanisms of biological systems, and then apply these knowledge to new to generate new technologies or medicine to benefit the society. So now I want to just spend the last 10 minutes or so to tell you a little bit about MBI Mechanobiology Institute. We are one of the a handful of research center of excellence in Singapore. So MBI was established just a little over 10 years ago. It's funded by the National Research Foundation and NUS, and the mission is to build leadership in novel scientific areas. And for us, it's mechanobiology, which is, like I said, it's an emerging branch of biological research. So current research at MBI is quite broad, and because we address mechanics on different scales, from the molecular scale. So for example, we want to understand how proteins can function as motors that move things around inside the cell. We study mechanobiology on the cellular level. We try to understand how cells move. For example, I showed you this movie of a white blood cell chasing bacteria. You know, how the cell actually navigate, how the cell have a very sensitive nose for that bacteria, and then move specifically in that direction, and how the cells communicate with each other in a multicellular tissue, or how the cells sense the environmental cues or properties. And then we have multicellular mechanobiology. How do cells come together to generate tissues of different shape, different properties, and how do these processes change during the aging process. And here we're also interested in how we can start from stem cells and to drive them into different kinds of specialized cells towards regenerating tissue. And lastly, we're very technology savvy, and so there's a lot of innovation in new kinds of technologies, from mechanomodulators to novel imaging platforms, because like I said, we really need to image at very high resolution, temporal and spatial resolution. So we always need to push the envelope of our imaging technology, and we try to come up with novel diagnostic devices. So these are different areas of our research. And one of our future research area, I already alluded to, is aging. So if we think about the cell as a machine, then just like machines that we see in our daily life, as they get old, they get damaged, or they suffer from wear and tear over time. But then the cell also has amazing ability to repair itself, to replenish. And force underlines both processes, because force can cause the wear and tear and damage, but force actually in biological systems also generates a signal for repair. And so what we think that aging process is a change in balance or loss of balance, so that there's more wear and tear than there's the ability to repair and replenish. So we want to understand how force drives both of these processes, and whether we can modulate this to slow down aging. And our scientists, just like our the subject of mechanobiology, our scientists are also very interdisciplinary. So we have professors from physics, from medicine, from biological science, from biomedical engineering, and we all come together. And this is where we're located. We're located in the T Lab building next to the engineering school. So here, scientists with different expertise work together in a big open lab structure. And we have our own MBI graduate program. This is a international graduate program that have with the goal of training interdisciplinary scientists. And this is what's unique about the MBI graduate program. We're not, focused on a specific area, but our students are exposed to a very diverse range of subject disciplines and learn how to use expertise from different disciplines to solve complex biological problems. So we normally have 6 to 8 students join every year. Currently, there are 35 students. We offer a full scholarship for 4 years that covers all tuitions, fees. We have offers our students a generous monthly stipend plus travel expenses. And so here's the basic program. We have courses in mechanobiology. And one element of our program that's really unique is the laboratory rotations before students starting their project. Our environment is an open lab environment. So it's a big open lab that everyone work together. So students learn a lot from each other and have the ability to interact with other researchers at different level, whether they're research fellows or even professors. So it's a very interactive environment. And so we don't like silo. We like to all work very closely together. And there are lots of other programs with our graduate students. So we have a MBI Young Scientist group. Actually, this week, they had their own international mechanobiology symposium where they had 150 participants from all over the world. This is completely organized by students and trainees. Our students come from also from very different backgrounds, from chemistry, life science, physics, engineering, mathematics. So if your undergraduate background is in, it's not biology, that's not a problem. And in fact, we like to have a really good mixture of our students because they can really learn from each other. The students, just want to say that students in our program are not just, learning, they're actually making cutting edge discoveries. So our students are very successful. They publish in these very high impact journals, and almost all students graduate with at least one, but often several of these high impact publications. And our students have won international awards, and they are now in, they all move on to either further more advanced training as postdoctoral fellows, or go into industry and other sectors where they can utilize their interdisciplinary knowledge. So some are in academia, whereas others are in industry, or even law, or other regulatory agencies. All right, so here's just a summary. These are what's special about our graduate program. I know I'm running out of time, and these are all the uniqueness about our MBI international program. One thing I probably didn't mention is that there's a great exposure to international science at MBI, just because our environment is very international. So if you want to learn more about our program, please visit our booth. Thank you.
Thank you very much, Prof. Rongli. We have maybe time for just one question, and okay. I think there was a question in the Q&A. Would you like to answer the question? It says, I found the MBI studies to be very inspiring. My research interest and experience sits in the mechanobiology and cell matrix interaction and in regeneration, especially antithesis and osteochondrial. How are these topics being studied in MBI? Are there some PhD projects on these topics?
Well, so I think these general topics is actually a perfect fit with what the interest of MBI. But so we study cell matrix interactions, mechanobiology and regeneration in tissue formation, and so this general area is a great fit. So I highly encourage you apply to our program. But one thing I wanted to say is that I would like to encourage you when you come into a graduate program is to keep a really open mind. Even though you study something that you're really interested in as an undergrad, when you come into the graduate school, it's going to be an eye-opening experience. My own story was that when I went to graduate school, I actually, just like you, I worked on a really interesting project as an undergrad, and I fixed my mind to work on that. Which was at the time was RNA splicing. But then when I actually chose the graduate program where there's a very strong, research, very strong research in RNA splicing. But then during the first year, when I was taking all these courses, exposed to all these advanced research, I completely changed my mind. I didn't even do a rotation in the splicing lab. I completely did something else. So I just want to encourage you to have an open mind, because you might find when you come into our program, you might find that there are lots of other stuff that you'd rather study.
Okay, thank you. Thank you very much, Prof. Rongli. I think we're running out of time.
Okay.
There's one other question. Perhaps you could answer by typing the answer to Prem. If not, then we'll address the question later.
Yeah,
okay.
that would be a question that's probably best addressed at our booth with our
Okay, thank you.
Okay.
Yes. So